Hello guys, I'm and ISC presently studying at Sri Venkateswara College of Engineering. In today, I am going to let you know about proteomics. Proteomics is a study of proteins and their functions within organisms. It involves the identification, characterization, and qualification of proteins as well as the analysis of protein interactions and modifications. Proteins is a rapidly growing field that has applications in many areas of biology and medicine. The techniques used in proteomics: mass spectrometry, two-dimensional gel electrophoresis, protein microarrays. Mass spectrometry, a technique used to identify and quantify proteins based on their mass to charge ratio. Two-dimensional gel electrophoresis, a technique used to separate proteins based on their isoelectric point and molecular weight. Protein microarrays, a high throughput technique used to study protein-protein interactions and protein functions. The use of proteomics in drug design. Proteomics can also be used to design more effective drugs by identifying the specific proteins that a drug should target and optimizing the drug's chemical structure to interact with those proteins. The approach, known as rational drug design, can lead to more potent and selective drugs with fewer side effects. The applications of proteomics: drug discovery and development, disease diagnosis and biomarker discovery, food science and nutrition. Firstly, drug discovery and development. Proteomics can be used to identify new drug targets and to study the effects of drugs on protein expression and function. Secondly, disease diagnosis and biomarker discovery. Proteomics can be used to identify protein biomarkers for diseases such as cancer and Alzheimer's disease. Food science and nutrition. Proteomics can be used to study the protein composition of foods and to identify allergens and other potential health risks. The future directions of proteomics. Recent advances in proteomics research have led to the development of new technologies and methods for studying proteins and their functions. Mass spectrometry, protein microarrays, and structural proteomics. Mass spectrometry. This technique allows for the identification and qualification of proteins in complex mixtures such as biological samples. Advances in mass spectrometry have enabled more sensitive and accurate analysis of proteins. Protein microarrays. These arrays allow for the high throughput analysis of protein interactions and protein function. They have been used to study disease pathways and drug targets. Structural proteomics. The field focuses on determining the three-dimensional structures of proteins. Future directions. Proteomics research is expected to continue to grow and evolve in the coming years with new applications and technologies emerging. Single-cell proteomics, proteogenomics, advanced data analysis. Single-cell proteomics. This field aims to study the proteome of individual cells, which can provide insights into cellular heterogeneity and disease mechanisms. Proteogenomics. This approach combines pre- proteomics and genomics data to study gene expression and protein function. It has potential applications in cancer research and personalized medicine. Advanced data analysis. With the increasing volume and complexity of proteomics data, there is a need for new computational tools and algorithms to analyze and interpret the data. Machine learning and artificial intelligence are promising approaches in this area. Limitations of proteomics. One of the main limitations of proteomics is the complexity of the data generated. The large amount of data produced can be difficult to analyze and interpret. leading to potential errors and inaccuracies. Another limitation is the sensitivity of the techniques used. Some proteins may not be detected due to low abundance or other factors, leading to incomplete data sets. Finally, proteomics research is often limited by the availability of resources and funding, as well as the need for specialized equipment and expertise. In this topic, we got to know about proteomics, a brief note about proteomics, the techniques in proteomics, proteomics in drug design, applications of proteomics, future directions of proteomics, and finally the limitations of proteomics. Thank you guys. This has been my presentation. Thank you, Anandal.